Hi everyone. In this video, we are going to talk about artificial synthesis of insulin. We'll introduce three different technologies in 1994, 2014, and 2020. Let's get started.
Hi, my name is Menghao, and I'm gonna introduce the early stage of research on insulin synthesis. There were mainly two methods for preparing synthetic insulin, the double-chain method and the proinsulin method. The two-chain method is the earliest successful technology for the production of biosynthetic insulin. The main steps of the method are as follows. Recombinant DNA technology. The genes for the A chain and B chain of insulin are separately cloned to E. coli using recombinant DNA technology. These genes are designed to be fused with beta galactosides, forming a fusion protein. Cell cultivation. The transformed E. coli is cultivated in a fermenter to produce a large quantity of the fusion protein. Since the fusion protein accumulates in the form of inclusion bodies with the cells, it needs to be separated after cell lysis. Inclusion body recovery. Inclusion bodies are recovered from the culture medium using centrifugation and other methods, and these bodies are rich in the target fusion protein. Protein purification. After solubilizing the inclusion bodies, the A and B chains are extracted after the fusion part is removed. The A and B chains are then chemically or enzymatically combined to form the biologically active insulin. The advantage of this method lies in its ability to efficiently produce insulin, but the overall cost is relatively high due to the multiple complex purification steps involved. The proinsulin method eliminates the need for two different fermentation and purification trains that the previous option requires. In this case, intact proinsulin is produced instead of the A and B chains separately. The picture shows the key manufacturing process steps. The E. coli cells overproduce triple-met proinsulin in the form of inclusion bodies, which are recovered and solubilized. Proinsulin is released by cleaving the methionine linker using CMBR. The proinsulin chain is subjected to a folding process to allow intermolecular disulfide bonds to form, and the C peptide is then cleaved with enzymes to yield human insulin. A number of chromatography and membrane filtration steps are utilized to purify the product.
Casting plants have been utilized to produce recombinant proteins because of their advantage of cost effectiveness, high quality protein processing, absence of human function, ease of production, and presence of ecology mechanically for post-tracing modifications. Initially, the human growth hormone was the recombinant protein product extracted from transgenic topical plants. After that, numerous different products have been developed from plants, such as hepatitis B virus surface antigen, antibodies, industrial protein, and milk proteins. These are Abrelian finale plants with one of the types of plants we can use to produce the human insulin. This is its way to produce the human insulin. The A earlier plants was transformed with the plant's Boolean expression vector PSBS4405. The construct handles a fusion protein, allowing human insulin OBHIN. Comparison requires human mini insulins with an unfluorotyping prototype like 27 mini insulin fused to the C returns to the a brilliant phenol 18 kDa oleucine pellet A. Recombinant human insulin has been successfully pressed and produced in oil seeds of plant Apparatus Philonia. This technology enrolled the target expression of insulin and in subcellular organic cell known as oil bodies that allows very high level of expression with easy recovery of recombinant insulin. Oil body are storage origins inside the oil seed, which consists of hydrophobic perfectly encapsulated by photophilic membranes and an outer wall of proteins known as the origin. Genetically energy oil seeds have been generated with recombinant proteins specifically targeted to oil bodies as origin fusion. Then the oil body are easily separated from other seed components by liquid phase separation, which reduce the number of cumulative steps required to obtain pure free insulin. It has been observed that the insulin accumulates of high level in transgenic seeds, about 0.13% of total seed protein. Recombinant insulin was derived from the oleucine fusion partner and merger with tyrosine deficits following oil body purification to yield a biological active insulin.
Hello, I will introduce the new technology in 2020, a PCR-based strategy for producing the human insulin. The first step is prepare the reaction system, including the template DNA, the primers, the DNTPS, and other components. After that, is the denaturation step to heat the reaction system to the 95 degree to separate the double-stranded DNA. Then, annealing to lower the temperature to allow the primers to bind to the single-stranded DNA template. After that, the extension at the moderate temperature, the DNA polymerized use the DNTPS as the raw materials to synthesize the new DNA strands. After that, we'll back to the denaturation step for 30 cycles to obtain a large amount of target DNA fragments. Then, the PCR products was purified using the DNA recovery kit. The DNA was isolated from the PT21B vector. Subsequently, the PCR applicant and the vector were digested with two enzymes. Then, the digested PCR product was ligated into the vector using the T4 DNA ligase. The vector carrying the human proinsulin was transformed into the cells to go to the expression and isolation. And the next picture, the inoculation for expression step. The expression of human proinsulin was induced by the addition of 0.1 to 1 millimole IPTG at an early exponential phase. Then after overnight induction at 16 degree, the cultures were transferred to the 50 milliliter cell star centrifuge tubes and spun down at 8,000 rpm for 15 minutes at 4 degrees. Then the pellet was resuspended in the 50 millimole ferrous hydrogen chloride. The crude protein extracts were sonicated for 5 to 10 minutes on the S until the cells were homogenized. Then step centrifugation and supernatrient transfer. The samples were centrifuged at 8,000 RPM for approximately 15 minutes at 4 degrees. The supernatrant was transferred to a clean centrifuge tube and stored at the 4 degree. After that, the conversion of the proinsulin was converted to the mature human insulin as a result of the autocatalytic cleavage. In the end, the concentration of the crude protein was concentrated using the cyclotolton American ultraside exclusion, the centrifugal filters. Above all, we'll go to the detection. The quality of the biosynthesized insulin was assessed through the protein sequencing, conforming a 100% similarity to the human insulin sequence. The biological activity was equated using an in vitro MTT-SC with the HEPG2 cells, demonstrating that the crude biosynthesized insulin exhibited a similar efficacy to standard human insulin.
Now, I'm going to introduce the industrial planning and forecasting for insulins. Here in the screen are two charts for market analysis. From the chart, we can get some information. Now, the first information is that global insulin demand is growing, which is very clear. The diabetic patients are increased from over 400 million in 2018 to over 500 million in approximately in estimated 2030. Well, from that state statistics, we can estimate the demand by virus to be 630 vials in 2030. From the right chart, we can see that the majority of main consumers are manufacturers of diabetic medicines, hospitals and clinics, and research institutions. They act as the bridge to connect diabetic patients and insulin. Secondly, let's look at the resource allocation. There are three major resources: that's human, material, and financial. Each resources are allocated by different ways, and in order to get different goals. Well, let's quick pass through this and get to the third part, the most important part. So it is by financial forecasting. We are not just planning industrial, but also forecasting our revenues and cost and do a break-even analysis. Here in the chart are some costs by different components. And apart from the cost, by consulting the market statistics, we can draw the conclusion that the average selling price will be $0.12 per megagram. That is a fair price. So our break-even analysis will determine how many insulins we will produce per month. So using the average cost, which contains fixed cost and variable cost. And we estimate the batches and list the equation to solve. We got is approximately 9 batches, which means 90,000 megagram per month. So we can meet the break-even point. That's all.
This is the end of our presentation. Thanks for watching.